Welcome to BioCode. In this video, we'll see how we can utilize Grep command on our computer, which is a very useful and very powerful tool that is provided in Linux and in various programming languages as well, such as Bash, Python, C++, Java, and etc. But Grep is also available on macOS and on Linux as well. It is unfortunately not available by default on Windows, so you can use any programming language to do that, but on Linux it is by default present. So Grep is a hugely popular tool that allows you to find or match patterns within a file on your computer. For example, in bioinformatics, I'm going to give a bioinformatics related example so that you can understand the applications of Grep. For example, if you have a example, if you have a genome sequence and you want to find out if out of all these protein sequences or whatever information that you have, you want to find out an exact single letter differences or single letter pattern matching within that file, you can do that as well. So I'm going to demonstrate something. I'm going to demonstrate various applications of that. But first of all, what I'm going to do is I'm going to display something very unique to you. So that you can understand the example. If you don't understand any of this right now, please don't worry about that, because okay, so this is not the file, because it is going to take some time before you understand what is happening on the computer. So first, suppose that we have a sequence.fast, this file, we do have this file, and we want to understand if any letter that is available related to in this file content is available within this file. For example, I want to see, don't worry about this command, I will explain everything, but I just want to give an example first of all. Suppose that I want to see if G letter is present within this file. I'm going to type in G, I'm going to type the full file name, then press enter, and it will pinpoint the exact locations where G is present within this file. It does not care about any other content, it will just pinpoint the exact location. So let's suppose that we want to see if gene is present anywhere, so it will pinpoint the exact location as well. So it will only display the content that is being matched rather than the content that is not being matched. So if you want to see the content that is not being matched as well, you can utilize the V command, and it will display the region that has not matched, and the region that is matched will not be displayed at all. But this was a very fairly simple example to display so that you can understand. But what I'm going to do is I'm going to retrieve a GTF file from NCBI, which is a human genome GTF file. I'm going to store it on my computer, and as soon as I'm done, we will extract this file in. I think we already have this file. Okay, so we already have this file. Let's get over it so that you can see. And this is a file. This is a genomic file, by the way, in GTF format. All of these are genes that are stored on MCBI and that are basically present within the human genome. So let's do more meaningful, and we will do it like head and file. So we will just display the, let's do 10. So this is a file that is stored, this is the content that is stored in this file, and what we are going to do in this case, we are going to find uncharacterized proteins, uncharacterized genes that are present within the human genome. So in any other methodology, I can give you an example. For example, if you want to do this, the same content, same procedure, you can do it on Excel as well, but it will take you huge amount of period to actually do that, because it is not recommended. You can do it on Python as well, you can do it on R language as well, but it will take a few lines of code to actually do that. But on Linux, we don't really have to do that. All we really have to do is use the grep command, and then type in anything that you want to find out in the genome.